I'd like to start by thanking the organizers for allowing me to tell you what we've been doing with Galaxy. And so a few months ago when we wrote the abstract for this conference, we were still calling our Galaxy instance FlowGalaxy. Since then, we've changed our mind. We're actually now calling it Import Galaxy because really it's going to be part of the Import ecosystem. So Import is a resource for scientists to store and share their data. This is mostly immunology-related data. We currently have, this slide is not up to date, and I apologize. We had a data release last week. We currently have over 200 studies that have been released. Those studies are clinical trials, transplantation trials, response to vaccination studies, these type of things. There's a lot of really cool things that you should go check out at import.org. There is actually over 35,000 subjects currently in all of those studies that are regrouped over 1,000 experiments. And those experiments have generated almost 1 million results. So again, those numbers are not up to date. So those results are actually a bunch of things. There's ELISA data, there's ELISpot, there's HAI, and today, and gene expression, which probably means more to you guys today than any of the other stuff. But today I'm going to be talking about flow cytometry. And so in the context of Import, we have something to store the data, to share the data. We wanted to give our users a chance to analyze their data also. And this is going to look familiar to most of you. We wanted something that's easy to use so that you can click and play. You don't have to deal with knowing how to program. Somebody else does that for you. We wanted something that's open source, something that has a history so that you can keep track of what you're doing. We wanted something where you could share workflows and data. And we wanted something that would support high-throughput analysis. So how many of you actually know anything about flow cytometry? Woo! More than expected, that's great. Okay, so for those of you who don't know anything about it, I'm gonna spend some time explaining the basics, and then I'll show you what we've been doing. Really it starts with your immune system. So your immune system is a bunch of different cells, cell populations that are derived from a hematopoietic stem cell. And all of the cells, all of the cell population differ from one another on a lot of things. But really what immunologists look at to define those populations is the marker that they express at their surface. So those are proteins that are expressed at the surface. And each of the populations of your immune system cells are going to be defined by a specific set of markers that they express or that they don't express at their surface. So you end up with cell population called like CD4+, CD3-, CD19 low, et cetera, et cetera. And it doesn't mean anything to us, but it does mean something to someone somewhere. Right, so how do we get to identifying in a given cell sample what populations are in there? So this is where flow cytometry happens. So you basically have, you raise antibodies to your markers of interest, you conjugate them to fluorochrome, you mix them with your cell sample, you feed that to a flow cytometer, which is a really cool machine when you think about it. So you have a single, you have a very thin capillary, your cells are going through it, single file, so one cell after the other. There's a laser that's shown through the cells, and the light emitted from that is captured by a bunch of different channels. So you have two things, you have forward and side scatters that gives you an idea of the size of your particle, and then you have all of the fluorescent channels that capture that information about what antibodies are linked to your cell samples. All of those parameters are acquired for each particle, so it's also called an event. And everything is saved in a binary format, which I'm not going to show you because it's not pretty, but if you convert it to text, you end up with something like this, which is a tabular format, and that we can actually do stuff with. So your typical workflow in flow analysis looks something, again, pretty familiar to, I'm assuming, most of you. You have a QC step, then you do some manipulation of your data so you can actually analyze your data, then you analyze your data, and then you get to the population of cells that you wanted to identify. So for QC, we use data from the flow cytometer, so you're looking at signal acquisition, dynamic range, flow rate, a bunch of things I'm not going to go into detail. Then you do compensation, which is basically correcting for overlap of fluorescence emission spectra. Then you do transformation, because again, your raw data looks like this. And that blob at the top corner here really looks like this. So you can't really do anything meaningful with this, so you need to transform the data. There's out there 5 or 6 different algorithms currently that people kind of fight about which one is the best one. But once you transform your data, you end up with something that you can actually work with. So the first thing you do is you clean up your data. So there's this blob in the corner, in the lower corner here. This is actually particle, like debris and dead cells. So you don't want that in your analysis, so you remove those. And then you have to identify in all those dots here, you have to figure out what is what. So this is where you do something called gating or clustering. So I'm going to explain gating first. And so really, a flow cytometry experiment is dependent upon the gating strategy. So you start your flow cytometry experiment thinking about how, when you get to your data, you're going to be identifying your populations. So you need to have a strategy in place before you start your experiment. And so gating is really a very long, painful, slow process where your poor grad students or undergrads or whoever you're not paying enough is squinting at a computer and basically looking at all of these different scatter plots and circling around clusters of cells in different dimensions to figure out which groups of cells express what markers. So for instance, here, if you follow the green, right, I don't have a, oh yeah, I do have a pointer. Okay, so you start here in that lower corner with the green population. Those are lymphocytes, which you know based on the size. This is side scatter, forward scatter. And you're going to look in the CD19, CD3 dimensions, and you know that in that lower corner here, they don't express CD19, they don't express CD3. So you're going to look at those cells, and then you're going to look in another dimension. Here, are they expressing HLA-DR and CD11? And if they do express both of them, then you know that those are dendritic cells. So you can see it can become quite complicated and convoluted and hard on a grad student who does the work. So another strategy is to use a clustering algorithm. There's a few out there, and so you basically end up with the same idea. Your clustering algorithm is going to digest all of your data, figure out what patterns of fluorescence intensity you have for each markers, define some population, and then go back through all of the data and assign each cell to a given population. So once you have all that, you've identified what your different subpopulations are expressing, you can go on to query the cell ontology to have an idea of what those cells are. Okay, so how do people do this? They can do it one of two ways. They can either do it locally by using a commercial software. The most famous one is called FlowGo. It's a commercial software, so it really depends on what they want to put in the software. Not necessarily very flexible if you have a very specific type of analysis in mind. You can also use R packages. There's a bunch of them that have been developed by very smart people, most of them on bioconductor. And if you don't want to do it locally, you can also use online resources. Currently, there's two that I'm aware of. One is GenePattern that maybe some of you know already. And there's another one called Cytobank. So GenePattern has a lot of cool things, but unfortunately, the flow cytometry analysis suite is not really up to date. And I don't know that it's actually maintained. Maybe it is, and if it is, I'm sorry. And then there's Cytobank. Cytobank is pretty cool, but it's also not free. You have to pay a subscription to use all of the packages that they have. And so in that context, well, you guys are convinced already that Galaxy is awesome, so I don't have to convince you, but that's where we wanted Immport Galaxy to go. So we wanted something, again, that's open source, that has a history, where you can share your data, you can share your workflow, something that we can keep up to date easily, something that where the community can contribute tools that they want to have. Okay, so this is what Immport Galaxy looks like. It will be hosted at importgalaxy.org. It's currently up. You can go have a look, but it's still in a test environment, so it's going to break probably. But it should be up at the end of September. So you can load up your data, or you can get data from Import. And once you have your data up there, so we have tools for pretty much every step of the analysis. So we have one QC tool. We have tools that read in FCS binary file. do the compensation, the transformation, convert it to text. We have a tool that has automated gating just for removing the debris, not for the rest of the gating. For that, we have a clustering algorithm called Flock. We also have a tool that compares the Flock output to all of your individual files. So this takes, some of those tools, I should say, take dataset collections as input, or your regular dataset. It doesn't really matter. Some of those tools are written in Python, some of them are R packages, some of them are C, so we have a bunch of everything. And to go back to the workflow, we also have a tool that queries the cell ontology and returns some idea of what your populations are. Okay, so I'm going to show you on YouTube, because this does not work. Okay, I'm going to show you the output of the overview of one of the tools. So this is an HTML format, and it's the output of Flock. All right, so it's an HTML format, right? So you have tabs here. This is supported by Bootstrap. And the first tab is the actual data that you feed into your overview. So it's a table. This is represented with data tables, so you can reorder the columns, you can sort, you can hide some columns if you don't want to see everything. You can then copy to your clipboard, so you get a tab-separated format. You can download a PDF or a CSV, and I'll go back to it. Then there's the overview plot, where you have all of those different scatter plots that I was telling you about earlier, and you can look at each of those individually with a third tab. So you select your markers that you want to see, you select if you want to see the points and the centroids or both, then you update your display and it works. This is generated with Plotly, so you can zoom in, you can zoom out, you can save as a PNG. You can also select the population that you want to see on your graph. So you click them, and then update your display, and then magic happens. You can also look at those same data in a 3D view. So you're selecting 3 different markers, just like before, then you update your display, and then you have your 3D visualization that you can play around with and kind of, turn in every direction. Just like the other scatter plots, you can select the population that you want to see. Then we have a parallel coordinate graph, which I think is kind of cool, where you can look at all the fluorescence intensities of each of your markers for each of the populations. So again, you can select the population that you want to see. You can reorder the axis if for some reason you think that the markers should be ordered in a different way. And you can select your population directly by brushing on the axis. So for instance, here I'm selecting CD4 plus, CD8 minus. And I can see at the bottom what populations are corresponding to those criteria. Then we have the cell ontology results. So we're using a tool called FlowCL for querying the cell ontology. So here are the results. So you have the cell ontology query, you have the number of results, the top result label, and the top cell ontology term. So if you click on the cell ontology query, you get the actual result from the R package. If you click on PDF, you get the PDF output from that package. And if you click on the cell ontology term, you're taken to the cell ontology browser that's hosted by Immport. Okay, and the last tab is the population score profile, which is what we're using to query the cell ontology database. And then once you've done all that, you can go back to your first tab, and you can use that comment column to change the name of your population, and that carries over to the other tabs. So now instead of having population 1, 2, 3, you can actually rename your populations. Okay, so let's go back to the slide. Okay. All right, so again, we want this to be a public resource for flow cytometry analysis, and we really want to support high-throughput analysis because it's really going towards that. Eventually, we do want to add to the toolbox because there's a bunch of other cool R packages that could really be in there. We also plan to do, we would like to do more work on data visualization. Everything is dependent upon funding, obviously. But because this is Galaxy and this is open source, we don't want to be the only one working on it, and we really welcome independent contribution. And I do want to point out that the QC package that we have It's called Flow AI. The person who wrote it published his paper in April of 2016. We emailed him. He was super enthusiastic, and he did all the work to get the tool wrapped and up in Import Galaxy. Okay, so with that, I'd like to thank all of the people in my team. The people from the Brinkman Lab have written all the R packages that I've been using for FlowCL and for visualization of some of the Flow Cytometry data. And with that, I'll take any questions. Yeah. Yeah, so the question is, why are we using Plotly and not Shiny? I think John should answer, because he's the one who made the decision. I think the short answer is that with Shiny, you'd need more work for it to work within Galaxy. We wanted something that was easy to manipulate.
Cool. Any other questions? I'm kind of new to the flow cytometry thing. About how big are the data sets that you're dealing with?
So that it depends on what you have. So in terms of text files, you can have something that's 20,000 event, which is it's 20,000 lines in your text file, but that can go up to millions and millions of events, depending on how much data you're generating. And if you're looking at 2 markers, you have only 2 columns, but if you're looking at 10 or 15 markers, if you're looking at site-of data, which is another type of flow cytometry. So it can get, short answer is it can get really big.
All right, let's thank our speaker one more time, please.